If you look at the back end of any major hospital laboratory today, you are basically looking at a marvel of modern manufacturing. I mean, you have these massive multi-million-dollar tracks using AI and, precise robotics to analyze clinical chemistry.
Oh yeah, the analytical phase is, it's completely industrialized at this point. It operates with just near perfect mechanical consistency. You've got analyzers calibrated to like fractions of a microliter.
Right. Running thousands of samples an hour.
Hmm.
But then you walk out to the waiting room and you see how we actually feed those incredible machines. And the fundamental process hasn't changed since the vacuum tube was invented like 75 years ago.
Exactly. We are still relying on a human being using their naked eye and their sense of touch to manually slide a needle into a vein.
Yeah, it's wild. We have this 21st century analytical automation that is just entirely dependent on a highly variable manual 19th century collection technique.
The vulnerability of the modern clinical lab is quite literally concentrated at the point of the needle.
And that paradox, that is the core of our deep dive today. So for those of you listening, whether you're in clinical ops, R&D, quality assurance, or regulatory affairs, we are unpacking the clinical benefit risk profile and the engineering behind a device that is actively rewriting the regulatory landscape.
Yeah, we are looking at Vitestro's Aletta, which is the world's first autonomous robotic phlebotomy device, or ARPD.
Right. And to really understand the magnitude of this, we have to look at the systemic healthcare bottleneck that actually forced the FDA to create a brand new regulatory framework just to accommodate it.
Because on August 19, 2026, the FDA granted de novo marketing authorization to the Aletta device. And, the de novo pathway...
It's fascinating, right, from a regulatory perspective, because it's reserved for things that are entirely novel.
Exactly. There was no legally marketed predicate device for this. The FDA couldn't just point to a previous machine and say, okay, prove you were substantially equivalent to this. They had to invent a whole new classification for automated vascular access systems.
And they didn't take it lightly.
No, not at all. The agency laid down really strict special controls. So they required comprehensive clinical performance verification, rigorous biocompatibility testing of all the fluid path materials, and extensive mechanical testing.
Right. They didn't lower the barrier to entry. They built this new, highly scrutinized pathway because the clinical void it fills is just so desperate. I mean, phlebotomy is one of the most commonly performed invasive procedures globally, but clinical labs are facing a devastating staffing crisis right now.
The numbers and the sources we're looking at are staggering. We're seeing annual phlebotomist turnover rates in U.S. clinical labs of up to 46%.
Wow, 46%.
Yeah. And when a phlebotomist leaves, the data shows it takes an average of 7 to 10 months to recruit, hire, and train a replacement.
Which is just a massive operational gap, and that cascades through the entire health system. Like if the lab can't draw the blood, the analyzers sit idle. And if they sit idle, emergency department boarding times skyrocket because, physicians can't discharge or admit patients without those diagnostic results.
It leads to severe bottlenecks
Mm-hmm.
and massive burnout for the personnel who are left trying to shoulder the load.
It's like having a state-of-the-art automated fulfillment center, but the loading dock is entirely dependent on one guy carrying boxes by hand, one at a time.
That's a great way to put it. For decades, drawing blood has been a strict 1-to-1 ratio. One healthcare worker, one patient, one manual draw. But Aletta completely fractures that constraint.
Right, we're shifting from this artisanal craft to a scalable, supervised workflow. Because under this new FDA clearance, a single trained clinical supervisor can oversee up to 3 Aletta devices simultaneously in an outpatient setting.
So you're tripling workforce capacity without compromising clinical throughput.
Mm-hmm.
And that's the operational leap that hospital leadership is analyzing very closely right now.
But, bridging the gap between a regulatory clearance and the actual physical engineering requires solving a massive technical challenge. My immediate thought, just looking at this concept, is about the unpredictability of human physiology.
Right, the anatomy.
Yeah. How does a machine safely perform an invasive needle stick on a human being without direct manual intervention? Like, how does it know where to go?
Well, the architecture relies on a synthesis of multimodal imaging, computer vision, and highly constrained robotic motion control. So the target acquisition sequence is built on active redundancy.
Okay, walk us through it. A patient puts their arm in the kiosk, and then what?
The machine initiates an autonomous scanning protocol using near-infrared, or NIR, light.
Right, and near-infrared utilizes specific wavelengths of light that are absorbed by deoxygenated hemoglobin. So that absorption makes the superficial veins appear distinctly dark against the tissue, which basically creates a 2D topographic map of the inside of the elbow.
Exactly. That 2D map gives it the initial trajectory, but it's insufficient for an invasive procedure on its own. So the system immediately combines that visual map with a 3D ultrasound sweep. High-frequency sound waves penetrate the tissue to determine the exact depth, diameter, and orientation of the venous structures. And then it deploys a really critical safety mechanism, which is real-time Doppler ultrasound, to analyze the localized fluid dynamics.
Because hitting a high-pressure artery during a routine blood draw is a severe clinical complication. The machine has to definitively know the difference between a vein and an artery before it even moves the needle.
Yeah, precisely. So the AI analyzes the acoustic Doppler signature of the blood flow. Venous flow is steady, it's low pressure, and it returns to the heart. Arterial flow is highly pulsatile, it's high pressure, and pushes away from the heart.
Right, you can actually hear that pulse on a Doppler.
Yeah, and the Doppler acoustics translate those flow characteristics into a mathematical signature. If the signature is pulsatile, the AI flags it as an artery and just completely walls off that area from the robotic trajectory.
I want to highlight a piece of practical engineering here that I found so clever because it solves a notoriously messy problem. Usually, to get a clear ultrasound image, you need that non-sterile sticky acoustic gel.
Right, which you can't use here.
Exactly. So Vitestro engineered a dual-use fluidic solution. During the scan, the system continuously sprays 70% alcohol onto the patient's arm.
Which is brilliant. The continuous alcohol spray acts as the immediate clinical disinfectant. So it preps the site to standard clinical protocols, while simultaneously serving as the acoustic coupling medium for the ultrasound probe.
It entirely eliminates the need for gel, keeping the whole process clean, sterile, and automated. But, and this is where I have to push back a bit, even with perfect sensors and elegant fluid dynamics, humans are unpredictable.
Sure.
Someone comes in dehydrated, their veins are flat, or they tense up and their arm shifts the exact second the needle touches the skin. What is the physical risk mitigation if the patient flinches or the anatomy just doesn't cooperate?
The fail-safes are layered incredibly deeply into the system. First, the AI operates under strict physiological parameters. So if the target vein doesn't meet exact mathematical criteria for depth and diameter, the machine refuses to attempt the stick.
It just stops.
It just halts the procedure. There is absolutely no scenario where the robot is, fishing for a vein under the skin like a frustrated human might do.
Oh, wow. Okay, so it's very conservative.
Extremely. Once the AI commits to a safe target, a mechanical arm inserts a sterile 21-gauge needle at a precise angle, between 10 and 30 degrees, executing with sub-millimeter precision. And regarding patient movement, the console is actually surrounded by high-frequency motion tracking sensors.
Okay, so if they twitch...
If the patient shifts their arm even slightly during the insertion phase, the system instantly triggers an automatic detachment protocol. It physically separates the needle from the robotic guide, retracts the needle into an onboard sharps container, and safely halts the session in milliseconds.
And beyond the visual and acoustic sensors, there's tactile feedback built into the robotics too, right?
Yeah, the device utilizes onboard load cell sensors. A load cell basically translates physical force into a measurable electrical output. So as the needle traverses the epidermis, the dermis, and subcutaneous tissue, the resistance profile changes.
Like it can feel how hard it's pushing.
Exactly. The AI is trained on the normal microresistance curve of a human vein wall. If the needle encounters tissue density that falls outside those expected electrical parameters, meaning it hits, say, unexpected scar tissue or a dense anatomical anomaly, it automatically pauses and alerts the clinical supervisor.
So the physical risk mitigation is really a trifecta. You've got active acoustic monitoring, passive physical load cell sensors, and strict software constraints.
Right.
But, engineering theory, no matter how robust, doesn't secure a de novo clearance on its own. QARA and clinical professionals need to see hard patient data. So how did this technology actually perform under clinical scrutiny?
Well, the foundational data comes from the landmark AIDOPT clinical trial, which stands for Autonomous Blood Drawing Optimization and Performance Testing. It was recently published in the peer-reviewed journal Clinical Chemistry.
And this was a pretty huge deal, right?
Oh, massive. It was a multi-center trial conducted across leading clinical laboratories, designed specifically to test both analytical equivalence and operational efficacy in a live outpatient setting.
So looking at cohort 1, which was 153 patients, the primary hurdle was proving analytical equivalence. because you can't just assume the robot handles the specimen exactly the same way a human does. You have to prove the molecular integrity of the blood isn't altered by the machine itself.
Right. So the researchers use passing Bablock regression analysis on paired samples. And for those unfamiliar, this is a highly stringent statistical model used in method comparison studies. It essentially plots the results from the human-drawn sample against the robot-drawn sample to ensure there's zero proportional bias between the two methods.
And they tested some highly sensitive parameters. I mean, they looked at coagulation markers like APTT and PT and citrate tubes. Because if the robot's needle creates excessive turbulence during the draw, it can prematurely trigger the coagulation cascade, which would completely skew those PT results.
Yeah, and they also analyze clinical chemistry markers like LDH and AST in serum tubes and total platelet counts in EDTA tubes.
Right, because if the robotic handling causes shear stress that ruptures red blood cells, those intracellular enzymes leak into the serum and your chemistry panel is ruined.
Exactly. But across every single parameter tested, the passing Bablock regression confirmed no statistically significant difference. The clinical equivalence was absolute.
Which is incredible. But that equivalence isn't just about how the needle enters the vein, right? It's heavily dependent on how the machine handles the tube after it fills, which is a massive pain point in the pre-analytical phase.
It really is. Pre-analytical errors account for roughly 60 to 70% of all diagnostic errors in a clinical lab. And a huge portion of those errors stem from improper mixing. I mean, manufacturer guidelines for tube inversions vary wildly. Citrate tubes require 3 to 4 inversions. EDTA tubes require 8 to 10.
And human phlebotomists are operating in these high-stress, high-volume environments. They frequently just eyeball the process, so they might shake the tube vigorously, which creates shear stress and destroys the red blood cells, causing hemolysis. Or, they might forget to invert the tube entirely, which leads to microclots that jam up those million-dollar analyzers we talked about earlier.
Yeah, but Aletta completely neutralizes that human variability. The robotic system mechanically performs exactly 9 standardized gentle inversions for every single tube it draws, immediately upon filling.
Wow.
It doesn't matter what color the tube top is or what the specific test is, the robotic arm executes 9 inversions at the exact same rotational speed every single time.
Think of it like CNC machining versus hand carving. The CNC machine doesn't get tired or rushed or distracted. It applies the exact same torque and angle on the 1,000th part as it did on the first. The robotic arm is applying that same industrial consistency to specimen handling.
And the trial data proved that this robotic consistency doesn't just meet clinical standards. It actively standardizes a critical pre-analytical step that is chronically mismanaged in manual workflows.
That leads directly into the efficacy data from Cohort 2 of the A.PT trial, which evaluated the system across 1,633 patients in routine outpatient conditions.
So the primary efficacy endpoint was the first stick success rate. When Aletta identified a suitable vein, it successfully drew all required clinical tubes on the first stick 94.5% of the time.
A 94.5% success rate is incredibly impressive. I mean, that places it on par with or even exceeding highly trained human phlebotomists. But the subgroup data is what genuinely demands attention from clinical directors.
Well, absolutely.
In patients classified as obese, with a BMI over 30, the robot's success rate actually jumped to 97.4%. And in patients with self-reported difficult venous access, or DVA, the patients who historically endure 3 or 4 manual needle sticks, the robot succeeded 92.7% of the time.
The machine is outperforming human phlebotomists in notoriously tricky demographics because it operates entirely outside the limitations of human senses. A human phlebotomist relies on sight and tactile palpation. But in an obese patient, the vascular structure is often buried beneath adipose tissue.
Right, you can't see it.
The human cannot see it and often cannot feel it. They're essentially relying on anatomical landmarks and making a highly educated guess.
But the robot's sensors don't care about adipose tissue. It's literally like having a submarine sonar system mapped onto the needle.
That's a perfect way to describe it. The 3D ultrasound waves penetrate the lipid layers seamlessly. Sound waves reflect off the fluid dynamics of the blood and the dense wall of the vein, presenting a crystal-clear acoustic boundary on the machine's imaging matrix.
So it's not guessing at all.
Not at all. For the AI, a deep vein in an obese patient is mapped with the exact same mathematical precision as a superficial vein in a thin patient. The guesswork is entirely removed.
And when you remove the guesswork, the safety profile naturally improves. Across those 1,633 patients, the adverse event rate was just 0.6%, and every single event was classified as mild, such as a minor hematoma or invasive vagal syncope, which is simply a patient fainting from anxiety. Which, let's be honest, is a physiological response that occurs in manual draws daily anyway.
Right, and the hemolysis rate, which is a critical quality metric for any lab director, was recorded at just 0.3%. This is exceptionally low. In fact, during the trial, there was a really revealing anomaly at Site 1 where researchers noticed a slight elevation in the overall hemolysis rate for the facility.
Oh, right, this was fascinating. So they conducted an ad hoc review of the manual phlebotomy samples being drawn by humans at that exact same site during the same time period.
And the manual human-drawn samples at that facility had a hemolysis rate of 6.9%. So the slight elevation wasn't a mechanical failure of the robot. The robot actually exposed a localized human error in sample handling practices at that specific clinic.
Wow.
Yeah, the machine's perfect consistency established a baseline that highlighted the flaws in the manual workflow.
Okay, so the clinical data verifies the engineering, and the FDA clearance verifies the regulatory framework. But integrating a disruptive medical device into massive health systems requires navigating the complexities of human psychology. I mean, you can engineer the perfect robotic arm, but if a patient refuses to let a machine insert a needle into their vein, the commercial momentum stops immediately.
Completely true. And the engineers at Vitestro anticipated that psychological friction. They designed the Aletta kiosk so that the mechanical tube switching assembly and the needle itself are completely hidden from the patient's line of sight.
It leverages the whole out of sight, out of mind principle. The patient never sees the sharp edge of the needle approaching their skin, and they don't sit there watching multiple tubes rapidly filling with their own blood.
And the data strongly supports this psychological shielding. In the NLPT trial, 90% of patients reported that the robotic draw caused less pain or an identical level of pain compared to their historical experiences with human draws.
That's huge.
It is. And when asked about future use, 82% either actively preferred the robot or had no preference between the robot and a human.
And Vitestro also ran a simulated use study in the United States at the Mayo Clinic, where 86% of patients indicated a willingness to use the Aletta device. So that high level of patient acceptance is really the catalyst for the commercial integration we are seeing right now.
The industry buy-in is very substantial at this point. Vitestro recently closed a $70 million Series B funding round, and the cap table includes strategic investments from the LabCorp Venture Fund, the Mayo Clinic, Sutter Health, and notably Dr. Fred Moll.
Right. And Dr. Fred Moll is the pioneer of robotic surgery. He co-founded Intuitive Surgical, the company behind the Da Vinci robot.
Exactly. When a pioneer of surgical robotics backs an autonomous platform like this, it signals a major shift. He's drawn direct parallels between the early skepticism surrounding robotic surgery and the current state of autonomous phlebotomy, really highlighting its potential to redefine an entirely under-innovated clinical workflow.
And they are moving aggressively to validate this in the U.S. market, partnering with Northwestern Medicine. Dr. Gregory Retzinger and Dr. Gary Noskin are actively working to validate how that 1-to-3 supervised ratio holds up under the chaotic, high-volume realities of an American outpatient center.
Yeah, and Dr. Thijs van Holten, who is one of the clinical chemists involved in the European trials, provided a highly relevant framework for this whole transition. He compared the integration of autonomous phlebotomy to the evolution of the supermarket self-checkout.
I love that analogy, because when self-checkout kiosks first appeared, consumer skepticism was incredibly high. Today, they handle the vast majority of standard transactions. But the human cashiers were not eliminated. The automated kiosks absorb the high-volume standard workflow, while the humans manage the complex edge cases and provide vital oversight.
Exactly. Aletta serves as the high-throughput kiosk for routine diagnostics. It allows highly trained human phlebotomists to step away from the relentless queue of standard outpatient draws. They can redirect their expertise to pediatric cases, extreme trauma patients in the ED, or just individuals who inherently require a human touch, while the robot ensures flawless pre-analytical consistency for the masses.
So wrapping up, the FDA's de novo authorization of the Aletta ARPD represents a massive operational paradigm shift. By merging multimodal imaging with rigid safety engineering, this technology is establishing a totally new standard of care. It mitigates physical risks, standardizes the chronically mismanaged pre-analytical phase, and offers a highly scalable solution to a global labor shortage.
And, the immediate focus is naturally on high-throughput, centralized hospital labs, as that is where the economic and operational pain points are most acute right now. However, considering the 86 to 90% patient acceptance rates, the next logical frontier for this technology is total decentralization.
Oh, that's an interesting thought. Because if the machine autonomously handles the complex imaging, the rigid safety protocols, and the sample standardization, you no longer need a dedicated phlebotomy staff at every single point of care. Imagine a near future where these autonomous kiosks are deployed in local pharmacies, neighborhood general practitioner offices, or even rural health clinics. It fundamentally shifts where, when, and how humanity accesses diagnostic medicine.
It really does. It takes the lab to the patient in a way we've never been able to before.
Well, for everyone listening, we hope this deep dive provided the rigorous clinical and operational insights you need to navigate this rapidly evolving landscape. Thank you for joining us as we explored the future of pre-analytical automation. Until next time.